The final hour, the fate of their semester. Got the whole stack, yeah, the pressure's on now. Flipping through the pages, take a careful bow. Mr. Marsh is coming for the final grade. Every single answer, every mark is made. Yeah, the pens are moving fast, a cinematic scene. Giving out the knowledge, keep the answers keen. Giving out the knowledge, keep the answers keen.
All right, science fans, this is Claire's Court, Common Task 2, Year 9 Biology, by me, Mr. Marsh. Welcome. I hope you got your headphones on, or you're listening somehow. This is a 45-minute exam. You can write in blue or black ink. You've got to answer all the questions, as usual. In all calculations, show out your working. You must show your working, and it is 47 marks. So it's about a mark a minute. For these papers, that's fairly typical for GCSE, about a mark a minute. So question number 1, read the top, always put your eyes at the top, answer the questions with a cross in the box, and if you change your mind, put a line through it like that. Some enzymes are involved in the breakdown of food substances. Draw one straight line from each food group to the products of digestion for that food group. So we have carbohydrates and we have fats, and then we want one line. So we've got lots of options. Fatty acids and glycerol, amino acids, glucose, starch and ethanol, and we're going to go for carbohydrate. Carbohydrate breaks down into glucose. There's my one straight line. Fat breaks down into fatty acids and glycerol. You just have to memorize it. It's in the book. There we are. Which enzyme breaks down fat? Carbohydrates, amylase. Another name for fat is lipid. So we've got the option of lipase, which breaks down lipids. So I put a cross there. 2 easy marks just from remembering from memory. Nothing to understand, just memory. Question number 2. Answer the questions with a cross in the box as again. At high pH values, the active site of the enzyme pepsin changes shape. We're dealing with an enzyme here. When an active site of the enzyme changes shape, the enzyme is specific, denatured, digested, or dead. Well, it's denatured. When we say it's broken apart or doesn't work properly, it's denatured. So, easy mark. DNA can code for amino acids in the active site of an enzyme. Okay. So, we've got an active site and the shape is specific. That's the point about an active site, like a lock and a key. Explain the role of the active site in the enzyme. So we know that an active site has to have a specific shape. An active site works on specific substrates, so it can form an enzyme substrate complex. So we're going to write about that. We're going to say the active site of an enzyme is a specific shape, because we're talking about it. So as soon as you hear anything about an active site, you go, oh, specific shape. That's really important. What else are we going to say about it? Because we've got 2 marks, you need to say at least 2 things. This allows it to bind to a specific substrate. The substrate is the thing that the enzyme works on. It could be fat, could be protein, could be starch, whatever it is. And in this case, it's DNA. So a specific substrate to make an enzyme substrate complex. So we've got an active site, which is a specific shape on an enzyme, if you remember the shape of an enzyme. That active site allows it to bind onto an enzyme substrate complex. So it bonds it together, weakens those bonds, make it easier to react, like a lock and a key. We talked about the uniqueness of that shape. And then you write that down. This helps to catalyze the reaction by lowering the activation energy. Seems pretty good so far. Now you only needed to get 2 out of these points, and I've got many, maximum of 2. I've got 4 potential marks out of that answer, and I can only get a maximum of 2. A student mixed, this is question number 4, turn the page. A student mixed 10 centimeters cubed of a solution of starch with some amylase, and amylase breaks down starch. A tube and a water bath at 25 degrees. You have to know these enzymes off by heart, you just have to. The student tested the solution for starch and glucose every 30 seconds, and figure 5 shows the results. So it says, was starch detected? And it goes, no. And then after a minute, yeah, glucose is detected. So that's weird. But then at 150 seconds, the starch has gone. And it says, why is it gone? What's one reason that there's now no starch at 150 seconds? So what is one reason for the result at 150 seconds? We could see that glucose, after a minute, starts to appear because the starch is being broken up into glucose. It's breaking it apart. Well, eventually, all of the starch will be broken up, won't it? All of the starch will be gone. So at 150 seconds, like a couple of minutes later, you are now at a point where all the starch has been converted into glucose, and because it's been digested into glucose, there's no starch left. Does that make sense? I hope it makes sense. You can't have something that you've broken apart. It doesn't work anymore. So that's why. Another student repeated the investigation with the same volumes of solution and the same temperature. So you can't use volume and temperature in this answer because they've already said it's the same. Give two other variables other than temperature and other than volume that you need to be controlled. So they've given the easy one. You go, well, enzymes work at a specific pH, right? So what about the pH? Did they keep that the same? Oh, that's probably an important thing to keep the same. How about the concentration? How much enzyme did you add? What concentration was it added at? That's important, isn't it? What else we got? How about starch was in the solution? Does it always the same starchy solution? What about how much you mixed it? Did you mix it the same amount each time? Did you mix one more than the other? Would that make it a fair test? They have to be mixed. What about the size of the tube, the size of the container? And what about the time intervals? Did you measure it every 10 seconds? Did you measure it every 20 seconds? Did you measure one for 20 seconds every 20 seconds and one for 10 seconds every 10 seconds? I mean, that wouldn't be. You have to keep things the same. Something that's likely to affect the results is what would count here. You only needed to come up with a couple of ideas. There's 5. Both students included a tube containing 10 centimetres of starch solution with just distilled water instead of the amylase. Well, we're testing if amylase breaks down starch. So by including the distilled water, we're going to prove that it's the amylase that does the breaking down. Because if it just broke down in water, the water would also break it down. That's called a control. So the control variable. So it is to check that the starch is being broken down by the amylase enzyme and not just by being in water. By putting it in water with nothing else in it, you're controlling for that amylase. It's a complicated idea, isn't it? But it's to prove it's the amylase that's having the effect. Talking about enzymes, a student investigated, question 5, the activity of a human enzyme at different temperatures. The student measured the mass of the product form after 10 minutes at different temperatures, and we got figure 5 shows the result of this investigation. So we have the mass of the product form in milligrams on this side, going up the side, and the temperature in degrees Celsius on the bottom. And oh, look at that shape. So it goes up to a point, and then it goes down, and it says describe the trend. So we've got to do some describing now, haven't we? You're looking at your graph, it goes up to a certain point, and then it goes down. We've got 3 areas that we could be talking about here, haven't we? So we're only describing. You could pretend you're an ant walking up the line on the page. So we've got the mass of the product, which is on the left-hand side. We could go, well, initially it goes up until it gets to 40 degrees Celsius. Look on your graph. It increases up to 40 degrees Celsius. The mass of the product then decreases after 40 degrees, doesn't it? But there's something else I noticed, and it's in the mark scheme. Look at the slope. This is interesting. That slope is not as steep before as afterwards. Afterwards, the slope is much, much steeper. We can make a note about that, and that would also get us a mark. So out of the 2 things you could mention, you could say, well, the mass of product that decreases sort of more quickly than it increased. So we got a difference in the slope. Anything that would describe it decreases faster than it increases. Look at the slope of the graph. You could get that because it's a describing thing. You can see it in the graph, so it's legitimate for a mark. Explain it. This is the why. Explain. There's your command word. So they want anything that's reasonable, scientific. What's the reason behind it? 40 to 60, what are we doing to the enzyme? Enzymes are quite fragile. If you get them outside of their optimum pH, get them outside of their optimum temperature, they're not going to be happy. They're not alive. They are a structure. They're a structure in the body. They're an enzyme. And if they're not so at 40 degrees, it's great. It's at an optimum. So we say there's a maximum product at 40 degrees because the enzyme at the optimum temperature is ideal for the enzyme to work. But as we go from 40 to 60 degrees, that enzyme starts to become damaged. The active site starts to denature. That means change in shape, to denature, to not be the same. And so it's not going to work anymore. The enzyme starts to lose its ability to form that enzyme substrate complex. And so the high temperature causes the enzyme active site to change shape and become denatured, and it starts to become less effective. Because it starts to become less effective, you're not going to make as much product. So you're breaking it apart, or you're bending it out of shape, and the enzyme doesn't work properly anymore. If it doesn't work properly anymore, it's not going to make as much as it did at 40 degrees when it was working at its optimum temperature. Enzymes have optimum temperatures, and enzymes have optimum pHs. You remember that. So here's the answer. High temperatures causes the active site to change shape, so it forms fewer enzyme substrate complexes, as I was describing. Turn the page. Oh, look at this. Love this. We've got a mitosis question. This is a root tip squash, is it not? Answer the questions with a cross. Right. Figure 4 shows cells from an onion root tip as seen from a light microscope. Okay. So here's a very lovely selection of cells. We've got cell Q and cell R. Need to know our phases here, don't we? Okay, here's the cell. Identify the stages of mitosis in cell Q and cell R. So is it metaphase, anaphase, telophase? In prophase, the nucleus starts to break down and spindle fibers appear. Right. At the end of metaphase, the chromosomes are lined up on the spindle fibers across the middle of the cell. The chromosomes copied are separate and move to either end of the cell. With the spindle fibers, this is anaphase. And in telophase, a membrane forms around each set of chromosomes to form nuclei. So let's look at this picture. By the way, once they come apart, the final part is called cytokinesis. That's when the cell walls in plant cells form, but this is also where you get 2 new cells. Let's have a look. Turn it sideways and it looks a bit similar to the book to me. Look at that. Spindle fibers lined up along the middle. Well, there it is. And there's it being pulled apart. No, oh, come on. That's anaphase, isn't it? That's a definite anaphase. And I think that one must be metaphase. It's lined up along the middle. Let's see if we've got that as an option. Where is it? It must be A. Well done. Metaphase and anaphase. It can't be any of those. It's got to be A. Metaphase and anaphase. You've got to know your cell cycle. Simple. Describe 2 processes that occur in cell during prophase. We've just looked at it. If you remembered it, it's in the book. Always look at the book, read the book. Chromatids condense, so they sort of coil up. When they coil up, they become more visible. That's really what happened. So that's a prophase thing. And let's think of the other thing that happens in prophase. The nuclear membrane breaks down, doesn't it? The nuclear envelope starts to disappear. That's the bit that holds the DNA, holds those chromosomes. It's in the book. You just have to memorize it. There's nothing to understand. It just says, describe the process. There it is in the book. Just describe it. Remember what it says. State the term used to describe the process which occurs after mitosis. We saw it. It's in the book, right-hand side. Cytokinesis, where you get the 2 cells. It's in the book, and there's the question. It's that easy to get marks in science. It really is. They can't ask you anything out of the book. Conversion now. So the diameter of this figure is 0.075 millimeters. And we've got to get it down to micrometers. So there's 1,000 micrometers. So we need to times 1,000. Let's have a look. 75. There we go. That's 1,000 times 0.075. It's just a times 1,000 question. There's 1,000 micrometers. Let's move on. We've got an osmosis experiment. Some students investigate water movement into plant cells. They've got a piece of potato, starts salt solution, and then the salt gets more and more salty. And then you've got the start, and then the after an hour, and they measured the mass gain. Notice that the percentage changes down the table, and they sort of lead you into spotting a pattern. You know, you could sort of guess what the pattern is. Look, it gains something, and then it's losing, and it's losing. Calculate the percentage change in the mass of the potato in the 20% solution. So that's the gap. And they give you the equation. Oh, that's fantastic. End mass. Okay, let's see. After 1 hour, we've got the end mass and we've got the start mass. There we are. So after 1 hour there, and the start is there. Let's put them in then, shall we? So the end mass we've got after an hour is 9.8 grams. And it starts at 10.3. So we'll just put those in then. So 9.8 minus 10.3 divided by the start mass times 100. Just put the numbers in. Just have to find them in the graph, in the results table. Hark at me. Put them in the results table, writing at the same time as doing it. So you put them in the results table. It's minus half a gram down. And we do the maths, put it into the calculator, comes out at 4.85. And if you want it to one decimal place, comes out at 4.9. So you get a mark for showing you're working, a mark to one decimal place, or the answer, doesn't matter. Right, suggest why calculating percentage change is more useful than calculating the change in mass. So this happens when anything, you've got things that are slightly different. If I lost 10 kilograms and you lost 10 kilograms, it would be a very different percentage of your body than it would be my body. So, you know, because of the starting masses of the potato are different, how are you going to compare like for like? It's better, it's more easy to compare them if you do it as a percentage change. If you lost 10 kilograms and I lost 10 kilograms, it would be a different percentage of our body weight, but it would be comparable then, wouldn't it, as a percentage? Because you're taking the mass, dividing it by 100. So it's an easy comparison if you start with different masses at the beginning. So because it starts with different masses, it's easier for comparison to do a percentage. And that's what you write. Where are we up to? Question number 8. Some substances move into and out of the cells by active transport. Active, energy, active. You need to put energy in. What does it say? Which is the correct description of the movement of a substance by active transport? Well, active needs energy, and you only need energy if you're going against something. So, oh, it's A. It's straight. It's given to us straight away. There we are. It's A. It's against a concentration gradient using energy. 9. So this is, you don't have to have done this experiment. You could just see that you've got jelly cubes in hydrochloric acid. There with a pink indicator. It becomes clear when it's in contact with an acid. It becomes colourless actually. Read the results of the investigation after the cubes have been in the cube for acid for 120 seconds. So let's have a look-see. The distance from the outside of cube B to the pink area was 3 millimeters. Fine. How far did it move in 1 second? It says calculate. So we're doing maths. It's calculate. 3 millimeters. It took 120 seconds. So in 1 second, it would be 3 millimeters divided by 120 seconds. And that comes out at 0.025 millimeters every second. See? Just maths. Didn't have to do the experiment. It's literally 3 millimeters in 120 seconds. How far did it travel in 1 second? And we're back to pore osmosis. A student wants to investigate the water movement into and out of cells of potatoes. And this time it's sodium chloride, but they've given you molar concentrations. Now you don't know what M means, probably in year 9. But we've got sodium chloride. A little bit more difficult because it's sodium chloride. But that just means salt. Why the test tube in the beaker only contains distilled water? Notice, same as the enzyme experiment. If you're just using distilled water, then it's likely going to be a control to make sure that it's the sodium chloride that has the effect. What else do we need to control in this investigation? Let's think about it. How are we going to make sure that it's a fair test? Well, temperature, that's going to have an effect, isn't it? Put it in boiling water, it's probably going to do something different. Is it an old potato? Are you going to use old potatoes and some that you got from the shop today? Is it the same variety of potatoes? Red potatoes, white potatoes? Does that matter? Or what the size of the pieces of chips that you're using? The length or the volume of the potato chip? Could you crinkle cut one chip? And how long you're going to leave them? That's important. Leave one in there for a week and in there for a day. The result's going to be completely different. Any two of those. So why did the 0.5 mole sodium chloride solution lose mass? It's a hint. If it loses mass, water's come out of it, right? if water's come out of it to make it get lighter because it's lost mass, then water has gone out of the cell membrane because there's more water inside the cell than outside the cell. So there's a higher concentration of salt, sodium chloride, outside the cell than inside the cell. And if that's true, then water's going to leave by osmosis. And we have to describe that. So we say there's a higher concentration of sodium chloride solution outside of the cells than inside of the cells. That's your first mark. And we know that water moves from where it is to where it isn't. So there's therefore a higher concentration of water inside the cells than outside of the cells. So guess what? So water moves from inside the cells to outside of the cells by osmosis, from an area of high water concentration to an area of low water concentration. That's the way it always works. You could say it the other way around. If you said there's a high concentration of sodium outside the cells than inside the cells, you could say there's a higher concentration of water, and you could say it backwards. So yeah, doesn't matter. Higher concentration of water inside the cell than outside, that works just fine. But the point is, water's going from where it is to where it isn't. Question 11. Figure 8 shows a banana plantation. So there's your sucker. After the bananas have been harvested, the old plants are cut down. The suckers then develop into mature plants, producing new crops of bananas. The tip of each sucker contains a group of cells called the meristem. Stem cell question. Describe the function of the meristem in the growth of a plant. You know this. You know this. You know that meristem cells are in the tips and the roots and certain parts of the plant where they can become differentiated. They can turn into different parts of the plant. Because meristem cells are undifferentiated, what they can do is then divide, and they can then turn into the other parts of the plant that you need, like the leaves or the roots or whatever. So what happens is they divide and produce more cells by mitosis, which allows it to grow and become differentiated. So a student took a sample of cells of the meristem to view under a light microscope. How would the student prepare a microscope slice? You've done this. You want a thin slice. Why do you want a thin slice? Something ideally 1 cell thick. Take the thinnest slice that you can of the meristem. You stain it. Doesn't matter what you add. You could say, I don't know, iodine or methylene blue, whatever works for you. Stain it and then put a cover slip on top. That's how you prepare it, isn't it? You put a cover slip on top. You've done it before. You take a very thin slice, you put a bit of stain on it, put a cover slip on, put it under the microscope. Ooh, look at this, a percentile chart. We like those, easy to read. So you've got a percentile chart for males aged between 2 and 20, you've got 95th, 90th, 75th, all the way down to 5th percentile. And you can see that they all follow the same pattern, but they grow at different rates. So describe, same as the other graph. You're just describing. What does it look like? What does it do? How does it change from 2 to 20 years? It's not dead straight, that graph, is it? Sort of about, what, 15 to 18-ish? We get a bit of a growth, but then it starts to slow down. Do you see that? And they'll accept anything between 15 and 18. So between the height definitely increases from 15 to 18. Goodness me. I mean, teaching some of you, I know that the height has suddenly started to increase a lot. But then it starts to level off. So we're just describing it. It goes up from 2, height increases from 15 to 18, and then after 15 to 18 it starts to level off. You're just describing. And then we have to do a calculate. As soon as they calculate, it's maths. Take an 11-year-old in the 95th percentile. So we take the 11-year-old and not 95th, sorry, 5th percentile. And the 95th percentile, and what's the difference? It wants to know what's the difference. So we go from there to there, and we go across. That comes out at what, 100 and... They give you a range, especially if it's a small graph. 132-ish? Say 132, yep. And that's 155. So what's the difference between 155 centimetres and 132 centimetres? You just do a bit of maths, don't you? That comes out as what, 23 centimetres? You had a range that you could answer within, because if you didn't quite read the graph right, you can still get the range. So what does 95th percentile mean? If I had 100 boys, the 95th percentile boy means that 95% of the boys in this case, or 95th percentile of people, will be smaller or shorter than that particular boy. So that's how that works. So if there's 100 boys, you line them up. The person that is taller than 95, and 5% will be and 5% will be taller than him. So he'll be taller than 95% of them, and only 5% will be taller than him. In 2012, 2 scientists were awarded the Nobel Prize for the research in stem cells. What's the benefits of adult stem cells being reprogrammed with the properties of embryonic? Well, we want stem cells because they are really useful to differentiate. But why would we use stem cells from an adult over embryonic stem cells? Well, we know that the nice thing is about stem cells is we can turn them into any type of cell, which makes them super useful because they can treat diseases that we don't have a cure for yet. That's important. And I think, if I remember rightly, stem cells can be produced any type of cell. They can be used to treat incurable diseases that we need new cells for. We just don't know how to get new cells. You can reprogram the cells back into stem cells. You can turn them into things. The other thing, when they come from the adult, they're not going to be rejected because they're from the adult's own body. So the adult, the patient, is going to not reject them. And that means that that you're more likely to be successful. In addition, you've got the ethical argument. You don't have to use embryonic stem cells. People have got religious beliefs about this. Some people might have ethical beliefs. Some people believe you're killing a baby because it's an unborn child, even though it's only a group of cells at the time. That's a valid argument as well. Some animal cells are stem cells, it says. Okay. How would you answer this question? So what's the function of a stem cell? Why do animals have them in the first place? Well, they are there to turn into all of the different types of cells that you need in the body. All cells in an embryo have to start off as undifferentiated, don't they? But they have to become different. So they start to divide by mitosis. They end up becoming different and specialized cells. So then they can create the rest of the body from that. So they could produce new cells. You could do it all through your whole life. You get damaged or you want to grow or replace damaged cells. Then a stem cell, it will grow from that into new cells, and that's the end of the test. Not that hard. How did you do?
This year, Mr. Marsh answers your exam paper.
The final hour, the pens are down, let's go. Flipping through the pages, putting on a show. Got the answer key, gonna break it all down. Every single question in this whole town. Gonna unlock the secrets, make the knowledge shine. Help you ace it next time, yeah, you'll be fine. Help you ace it next time, yeah, you'll be fine. Yeah!